Thank you, Dr. Simano. אני מנסה גם... יופי. כידוע, יש עוד כברת דרך לפנינו. תהליך פיתוח תרופה משלב הרעיון במעבדה ועד לשלב המימוש והשימוש הוא תהליך ארוך ויקר. העובדה שמספר חברות נמצאות כיום בשלב כה מתקדם של ניסויים קליניים ופיתוח תרופות גנטיות מאפשר לנו לחוש אופטימיות זהירה. המרצה הבאה, דוקטור מדלן בילטר, מייצגת את חברת סרפטה. דוקטור בילטר הינה חוקרת בכירה במערך הרפואי של סרפטה באירופה החל מ-2002. היא מביאה איתה קרוב ל-25 שנות ניסיון בתעשייה הפרמצבטית. דוקטור בילטר עבדה בתפקידים שונים בתחום הרגולציה הרפואית ובתחומים טיפוליים נוספים. דוקטור בילטר, פליז.
A little bit, so probably this is better. So good morning, everybody. It's a pleasure being here. Thanks so much for the invitation and the nice welcome words. I didn't understand it, but I guess I know what you said. It's really a pleasure being here the second time in Israel to present on behalf of Sarepta. And this time it's for me the first time to present on gene therapy, to be honest. So, as you heard, I'm the head of Medical Affairs Europe, and I call it now Europe Plus because officially I'm also now responsible for Israel and the Middle East. Having said that, I just want to... Sorry? The right one? Okay. No, that's not mine. Wait a minute. That's wrong. No, that's wrong.
No problem.
Cool. Sorry about that.
No, we're going backwards.
It's not only me. You're going the wrong direction. Here we go. So, wait a minute.
To the right.
This one, right. Okay, so as a disclaimer, none of our gene therapy is actually approved in any country yet, so we're still in the development of those countries, so whatever I'm telling you here is really on an educational purpose. We think we are armed with scientific, robust scientific evidence to fight for rare disease, and we are in a daily fight. So really our mission at Sarepta is that every day is another 24 hours to stand up for patients, advance technology, challenge convention, and drag tomorrow into today. That's a really, really high mission. And being a small company, what do we have in our hands to fulfill this mission? Now, as we speak, we are expanding into the globe. We established Sarepta back in 2013, and it's based in Cambridge, Massachusetts. And we opened a research facility 2 years later in Andover. Now, having said that, we are globally committed, a European headquarters based in Switzerland, where I'm also based. And we opened in LATAM back last year, and this year another subsidiary in Japan. It's very important, and we just heard it from the colleague from Pfizer, that we are able actually to produce our drugs. So in collaboration actually with Nationwide Children's Hospital in Columbus, Ohio, where we now also have a center of excellence for gene therapy, we also opened recently a manufacturing center of excellence in Burlington, Massachusetts, because we really want to be ready once we are going to be in phase 3, and I talk about that towards the end of my presentation, that we have enough drug to deliver and also when we go to the market. It's important that we have, of course, colleagues working around the globe and being based in the field. So we're not only having field-based people in the US and LATAM, but also in many European countries. And here in Israel, we have a true partner, TrueMed, which is here in the room. And I'm happy to say we are happy to have them on board as well. So in Srpta, we're not only working on gene therapy, we have an armamentarium of technologies. Our goal is really to develop a treatment for 100% of DMD patients, and so gene therapy is an essential part. But as you know, we also have the RNA target therapies, exon skipping, using PMO and the second generation PPMO. I guess you all are aware we have one approved drug in the US, but also here in Israel. Israel is country number 2 where we got approval for Exondys, which is eteplirsen for patients amenable to 51 exon skipping, and more to come over the next couple of months or year, depending on where we are in the world. We are partnering with a lot of academia, and we are partnering also with Genethon. Geraldine will talk about the Genethon development, but we also partnering with Duke University in the US on CRISPR-Cas9 technology. And with the partnership of Cherry Mandel Nationwide Children's Hospital, we are really have a great partner for gene transfer, microdystrophin, and also we have with GOLD-GT2, which we hear about later on in another presentation. So this is our pipeline in Duchenne, and you can see overall we have 15 programs running in parallel, which is quite a lot. So we have a couple in RNA technology, and today I'm gonna focus on microdystrophin, the nationwide compound. I'll come back to that later on. In addition to DMD development program, we also work with gene therapy in Pompe, Charlie-Marie Tooth, MSP, and other neuromuscular diseases, and so we have huge work to do. In general, this is our scientific approach to gene therapy. And I don't need to go into detail because actually Joël nicely described the gene therapy in general, but I just want to emphasize our approach. So we are using actually the FDA definition of gene transfer, where we add a good copy of the defective gene and express in the relevant cells. So we have the gene. We have actually the gene of interest, and of course in DMD it's the dystrophin, and we bring a modified, the transgene into a modified virus, AAV, and in our case it's AAV7, bringing it into the body, and with the help of the promoter, we heard about the promoter, it's really expressed in these cells of importance, like the skeletal muscle cells, diaphragm, and cardiac muscle. There are 3 essential components. We already heard about that. So we really need a viral vector. The vector, the selected vector, must deliver the transgene to target cells with minimal immune response. The minimal immune response, we heard about that before, is very important. The promoter, we have chosen a specific promoter that really drives expression in skeleton and cardiac muscle. And last but not least, the transgene must produce a functioning but shorter protein of interest. And of course, in our case, it's microdystrophin. Again, the adeno-associated virus is a delivery vehicle for gene therapy. It's really, really widely used. There is a lot of experience with this virus. More than 200 studies have used this virus, not only in DMD, but also in other disease areas. Important to be mentioned here that it does not integrate into the host genome. And this is a very important point because it does not alter the genome. The transgene, again, as we said, it is the full-length dystrophin is really too big. It has roughly 14 kilobytes, and the capsid or the vector only can host, or actually we only can package a small amount in there, about a third. So what we need to do is really shorten the transgene. So I'm happy to share with you now a couple of preclinical, but also later on some of the clinical data from our phase 1 trial, which is actually completed. But let me first go into the preclinical data. As I said, what we need to do is to have a microdystrophin and construct the microdystrophin. We already heard that the full dystrophin is huge, around 14 kilobytes, so we need to shorten it, and we know we have a couple of active sites which we need to consider to put in. So our microdystrophin looks the following. We have chosen to go for 4 active sites. I know the colleague in Solid said 5. So we have actually the CR and the CT site, and then we have 3 hinges, 1, 2, 4, and R1, 2, and 3, and R24. So with that construct, we are convinced we have a good microdystrophin that works in DMD. The vector has a robust affinity for muscle. It has a relative low level of pre-existing immunity, and this is a very good point. The promoter is specific for skeleton and cardiac muscle, and I'll show you later on some data on that. And it drives really expression in cardiac muscle, which is of utmost importance. And then, of course, the microdystrophin transgene is a protein that is functioning, a truncated version, and it contains spectrum-like repeats 2 and 3. So this slide shows you a nice study where, and this is actually in mice, in the MDX mice model, where we have chosen the wild type, then 3 different doses. And what you can see here on the slide is fiber expression, dystrophy-positive fibers in different muscles. So we have like gastrocnemius, quadriceps, psoas, and so on. So there is a dose-dependent activity, and you see that the highest dose here provides the best results. So we have the highest dystrophin-positive fibers in percentage in the highest dose. And based on these preclinical findings, we also then moved into the clinical studies with high dose. Looking at other results, of course, we all know that creatine kinase is very high in DMD. We looked at the CK reduction, but also muscle strain, and here as a example, specific force in the tibialis anterior muscle, and also eccentric concentration. There is a significant reduction in CK. It's not as low as in wild type, but it's a significant reduction, and an increase in specific force, and the eccentric concentration is also increased. Now, from a preclinical point of view, in summary, we think we have a construct with our structure AAV7, MCK7, and microdystrophin, and we will apply our learnings from the preclinical into the clinical space. So preliminary preclinical results show that the vector is transducing efficaciously in all muscle types. Preclinical studies demonstrated 120% cardiac expression, which is very interesting and very promising. We have a low pre-existing immunity for the vector. And again, as I said, the promoter allows for cardiac and skeletal transgene muscle expression. And now I'm happy to present some of the clinical data, as I said. Now, based on the preclinical data, when we set up the phase 1 study, the study 1, which was fully run at Still Nationwide Children's Hospital, we had some expectations for this trial. So we wanted also to show now in human beings, in boys, that the vector is efficient inducing to all muscle types, that the promoter really is selective for cardiac and skeletal transgene muscle expression, and we have a widespread microdystrophin expression in all biopsied muscles. Of course, we won't have a biopsy in cardiac muscle. From a clinical perspective, also reduction in CK as a surrogate marker. And last but not least, and this is very, very important for a phase 1, which actually is the main endpoint, we want to show that this construct has a favorable safety profile with no unexpected immunological response. Could we fulfill this expectation? Well, I'm going to show it in a moment. So to let you know, this is a trial design. So this trial was supposed to have 12 subjects. Basically, actually, we have 2 cohorts. Cohort 4 is fully enrolled and in the follow-up phase. Inclusion criteria, of course, confirmed DMD mutation. And as a prerequisite, we have to have negative antibodies for the vector. That's important. And this is measured by an ELISA technique. Patients had to be in stable steroid dosing for at least 3 months. And daily steroids started 1 day before treatment for at least another 30 days. As I said before, we have chosen a high dose, 2 × 10^14 Vg per kilogram. And it is a safety study, so the primary endpoint was safety, and secondary endpoints, of course, change in microdystrophin expression pre- and post-treatment as measured by immunohistochemistry and Western blot, and a couple of functional outcome measures like 10-meter time test and so on, and also at 1 year, a cardiac MRI and other functional tests. I did not show you actually the microdystrophin expression in the mouse model, because I'm happy to show you here the results from the 4 patients that were enrolled in the study 1. So on the left-hand side, you see a normal control, and this is the microdystrophin expression measured by immunohistochemistry. Subject 1, 2, 3, and 4. And below, on the lower level, you see the post-treatment results. It was a biopsy, ultrasound guided needle biopsy in the gastrocnemius. And there is actually a high intensity and also a high percentage of dystrophin-positive fiber in the 4 patients. Intensity in the average 96% and more than 80% percentage of dystrophin-positive fibers. The results in the animals were similar, but of course, and we also have shown that in the animals in the cardiac muscle, but here we only have skeleton muscles. When you look at Western blot, we also have Western blot of all the 4. It's important to be mentioned here that Nationwide Children's Hospital actually adjusted the Western blot for fat and fibrotic tissue. By doing so, they could show a mean expression of 95%. When you compare to the Srpta technique, which actually is used in all our other trials with the RNA technology, PMO, PPO, that's lower because we do not adjust for fat and fibrotic tissues. It's important to mention here. The 4 boys treated, fully treated and extension now, they are actually followed up for 48 weeks, and then we also will go into a long-term follow-up up to 5 years. The baseline level here from the CK you see are between 20 and 35,000. The numbers, patient number 3, actually he did not follow the protocol, so before the baseline measurements, he was doing quite a lot of exercise, so CK level were actually, it's kind of an outlier. Now, the CK values really dropped significantly over time, which you can see on the right-hand side. The next slide is a very important one because it is summarizing the clinical data we have as of today. It is the last assessment, and of course, as you can see, boy number 1, the assessment was at day 270, boy number 4 at 90 days, and that's of course because after the first boy we had to wait a couple of weeks until we could dose the second one due to the safety efficacy endpoint. Now, overall, when you look from baseline to last visit in all the boys, you can see improvement across the measurements. So there was an improvement in the average NSAA, time to raise, the 4 stairs up, this 100-meter walk measured in seconds, 10-meter, and also the CK levels, which I have shown you for 3 months, but now here I have the last assessment. From a clinical perspective summary, all the boys are doing fine, and I'm really happy to say that. So basically, if they're doing fine, I could stop here. So we have seen clinical improvement, actually, as I just showed you in the last slide, in the clinical assessments. We have shown dystrophin expression in ICH and Western blot and all the other biomarkers as well. And as I said, these patients are going now into a long-term extension. Now, what is the future? I just showed you the nationwide children's study, trial 1. As we speak, we are enrolling in 102, which is a Srpta-run trial where we aim to have 24 patients in the next couple of months. Those 2 studies, are supplied with research-grade supply. And I'll come back then to the next one. So study 102, which I just said is running right now, we have 12 patients enrolled out of the 24, is a 48-week randomized double-blind placebo-controlled trial with systemic gene delivery, the same dose as we had in study 1, using our SRP-901 with a 96-week extension. On the right-hand side, you see the study design. It's really important to mention here the double-blind placebo-control phase is 48 weeks, and then the patients on placebo actually switch over to Verum, so they are going to receive the infusion with SRP-9001 at week 48, and then are followed up for another 96 weeks. The objective really is to assess the safety and efficacy of 9001 measured by biological and clinical endpoints, and we have the 2 parts. We have a 1-to-1 randomization, placebo versus Verum, and then, of course, we sit. The patient populations, as I said, is 24 patients, age 4 to 7. They have a mutation between 18 and 58, and the dose is the high dose as came out from the preclinical studies. We are currently planning a phase 3. We don't have yet the full clinical development. We have some thoughts, and I'll come to that in a moment. But the next study will be a phase 3, where we actually are going to use commercial-grade supply, and that's why I have built up this manufacturing in Massachusetts. This will be a global study then, because currently we're running the studies in the US, but the confirmatory phase 3 study will be a true global study. We are aiming, it's going to be also a double-blind placebo-controlled trial with switch over, and of course we are adding additional endpoints like patient-reported outcomes and so on. Over time, Srpta aims to enlarge the population, going into younger, but also going into older patients, and actually also broaden the mutations. So that is the objective, that by the end of the day we have a comprehensive development program that hopefully then can be accepted by FDA and other regulators for approval of our gene therapy. Thank you for your attention. Have a question?
Why you focus on the 1858 and do you have a plan to include it?
Yes. As I just said, we are in the second phase 3 study, which we have good thoughts, but not the final approved outline. We will have cohorts where we go on one side younger, older patients, and enlarge to the full. That would be the second one, yes, yes. So we are focusing on 18 to 58 right now to have a 1 to 17, but also up. There are patients not expressing at all any dystrophin, so we wanted to have a cohort that, how should I say that, that is more specific, that we really know what we're getting out somehow.
Is there a decay of the expression? Does it go down the dystrophin? Is there a reduction of the dystrophin level with time?
It's too early. So I don't have that data. Yeah. Yes, so we had, like 2 weeks ago, we had 12, so half of the target is 24, and we have now 12 patients in that study. It's what? The range of dosing? We have the high dose. Yeah, we have the high dose. It's 2 times. Oh, as the patients come in, now we don't need to wait. We had to wait when we had the phase 1. We had the first patient, and then we really waited to make sure it's safe. And then we administered to the second patients, and then